And this is only a fraction of the Excel chart. There are 20 monocounts, and this is 3 microorganisms. And these are the actual counts of how many proteins there are. Right next to it is just a percentage of how many of the actual counts. So it's easier to read than the actual count. The data that I gathered, and I didn't want to show the other Excel chart because it's 400 different pairs. So this is just like a brief what my data looks like when I finalize it. The data I gather were given to all the graduate students and the professor to be used, and they would use this data to analyze and interpret. And that's it. And I would like to take my acknowledgement to my Professor Virgil Reddy Vijay, my co-interns, and Dr. Stas, Harvard Human Society, and anyone who contributed to computer technology. Thank you. Any questions? Yes? Okay, procedure slide. Okay. Proteins, yeah, sorry.
So what is the significance of your work? Why are you doing this?
The real-time application. Because these enzymes or proteins, they function well in high temperatures or really extreme high temperatures, and that they're with the marketing community, they want to create dishwashing agents out of these enzymes so they could work well in these high temperatures.
But why are you using bioinformatics to address that? Why are you using Perl?
Perl? Well, it's the language that my professor preferred to use.
Yeah, but why are you doing it?
Why? Me, personally?
Maybe it's a good idea to go and ask that question. What is the objective of your experiment? The ultimate objective of your experiment?
Okay, yes?
You would express is there any significant factor in leucine? It was one of the amino acids that was there present much more than any others. Is that going to be useful?
Yeah, they want to see patterns within. First, there are 24 extremophiles they're studying, but there are 6 in each category. So they want to see a pattern within the 6 microorganism under the example of category extremophile, mesophile. So let me go back to my chart. So these are the 6 mesophile organisms, and I bold them because I think they're significant to how they work or how they significant to their survival in extreme temperatures. Question? Okay. Yes?
In your range is the number of proteins, right? How do you calculate the number of proteins in the data?
I use a program. Well, I grab data from the NCBI GenBank, and I look, and when I have, it's in a text file, so it's a long sequence, and I have a program. Well, it's a computer program, and I run it in a way where it reads the text files and it counts the individual letters, and then it prints it out. So it prints out like a text file, another text file with like the amino acid and the number next to it. Does that explain it clearly? Okay. Okay.
How do you exactly calculate the number of proteins? Like you have to that number.
How do I exactly calculate?
It says 3,569 or something, right?
Yeah, there's a program that runs it and reads it. I know it's really vague, but it runs.
That's a good question. You should look into it. The computer can do everything, but you have to know the theory behind it.
It's the algorithm that's placed in the computer program that I wrote that reads it, and it